Okay, so basically, the basic working principle behind DNA vaccine involve the use of a DNA plasmid that encodes for a specific protein that originated from the pathogen in which the vaccine will be targeted. As this DNA inserted into the cells, it will be translated to form antigenic proteins. As this protein is foreign to the cells, thus it will trigger the necessary immune response raised against these proteins. So in this way, the DNA vaccine provide immunity against that particular targeted pathogen. So here is the general step involved in synthesizing DNA vaccines based on the DNA recombinant technology. So firstly, the gene of interest, which is the gene that is encoded for protein antigens, is isolated, and then this gene is inserted into an expression plasmid. So after this process, you will have this plasmid with a foreign gene. So once constructed, the vaccine plasmid is transformed into the bacterial cell, as you can see here, where the bacterial group produce multiple plasmid copies. So this is where the process is that we want to amplify the plasmid DNA. And the next step is that the plasmid DNA is then purified from the bacteria by separating the circular plasmid from the much larger bacterial DNA and other bacterial impurities. So, as part of the end products, is that this purified plasmid DNA is used as the DNA vaccines. So here are some of the advantages of DNA vaccines, which is it is capable of eliciting both humoral and cell-mediated immune response, and it produces a focused antigen of interest, which means that only a specific parts of the protein that are known to be antigenic will be produced, and it can develop a long-term immunity, and it is safe, which means that once we focusing to producing a specific protein antigens, we eliminate any other materials that can cause autoimmune response, like the case in by using the whole microorganism as the vaccine antigens. And due to the rapid development of the DNA recombinant technology, the manufacturing of large-scale DNA vaccines is feasible and it's quite cheap compared to the conventional vaccine antigens. However, it comes to several disadvantages as well, which is, for example, one of the obvious one is the DNA vaccine is limited to protein immunogen only. For example, some of the bacteria last times we know that we need the carbohydrate that can act as antigens, but for DNA vaccines, it is limited to synthesizing the protein-based antigen only. And lower efficiency in large animals and humans. Why I mention the lower efficiency is that we know that when we develop vaccine based on the use of whole microorganisms, there are so many danger signals from the bacteria or from the virus that can trigger the immune response. But with DNA vaccine, it's a really specific subunit base of the antigens, which is not strong enough to trigger the immune response. So that's why, for example, as you can see in the COVID-19 vaccines that utilizing the DNA or RNA based vaccines, we need a delivery systems, and in their case they use a solid lipid nanoparticles as a mean to protect the DNA or RNA from being degraded and also to trigger the necessary immune response. And there's a necessity for repeated on multiple dose because in principle the DNA vaccines is not as strong, as efficient as compared to when you use or giving or present the whole microorganism to the antigen presenting cells. And also it takes some time for the antigen to be processed and immune to produce the desired protein as the antigen. So, but what about mRNA vaccine? It's quite a similar pathway on how you can produce RNA vaccines, but how it will be transcribed and translated in the body will be different. So we know that two of the licensed COVID-19 vaccines, they are based on mRNA, which is the one from the Pfizer and Moderna. So basically, messenger RNA, or denoted as mRNA, is genetic materials that tells your body how to make protein. And this protein, for example, the spike to protein is the protein antigen that can be used to trigger the immune response. So basically, the in vitro synthesis of mRNA starts with the cloning of the target antigen into a DNA plasmid, which is quite similar approach on how you want to produce a DNA vaccines, and its subsequent linearization, even though you can still use a non-cell based, which is you can use a PCR to synthesize the mRNA synthetic vaccines. And after the transcription, the DNA template is removed using specific enzyme, and in order to increase the mRNA stability and its translation efficiency, the transcribed products will be enzymatically capped at the end of the synthetic sequence. And finally, the mRNA product is purified to remove any remaining DNA template. So here, what are the main difference between RNA and DNA vaccines? One of the obvious difference between mRNA and DNA vaccine is that compared to plasmid DNA, which must enter the nucleus of a cell, the mRNA only needs to be present into the cytoplasm, which means with an mRNA vaccines, you are one step ahead of a DNA vaccines. Thus, mRNA vaccines will never enter the nucleus and could not alter the recipient's DNA.